Hey, Chris.
Hey, Andy, how you doing, bud?
Good, good. How you doing?
Doing fine. Happy New Year.
Yeah, same to you, same to everybody on the call. John Flak, how are you?
Good. How about you?
I'm good. Good to see you.
Should we get started or give it another minute?
Yeah, no, I think we'll go ahead and get started. Welcome to 2022, everyone. I hope everyone had a nice holiday break and is energized for the new year, despite the obstacles that are immediately before us. We have two excellent talks today, Ebony Carter and Jonathan Flak. Before we get started, I have a couple of very brief announcements. The schedule for 2022 has 3 sessions today with Ebony and Jonathan. On February 2nd, we were to have had Mike Stitzel and Zach Knight. Unfortunately, Zach won't be able to make that particular day. He has a conflict. And we're going to do something different on February 2nd as a consequence. We'll have Mike Stitzel give his talk. And then we want to invite everybody that has been coming to this call to take part in a discussion of future pathways activities. And Chandra will lead that discussion. I'll be involved as well, and others from ADA. And we'd love to have input from everybody on this group about how to really continue this program from a scientific and from a programmatic perspective. And we'll finish our current schedule on March 2nd with John Campbell and Daniel Saradini speaking. But we are hopeful that our discussions on February 2nd will result in additional activities after March 2. So we would like to ask everybody on this call to go ahead and pencil in, put a hold on Wednesdays at 2 p.m., the first Wednesday of every month, because we hope to be able to bring forward some newly planned activities for the sessions after March. So the first Wednesday in April, May, and out to June. Another reminder that I have, and you should have, everybody that's an awardee on this call, a Pathways awardee should have received an email from the ADA requesting submission of abstracts to the ADA scientific sessions. And as I understand it, that's a mandatory thing. So we hope that everyone will do that. There's been wonderful science that we've listened to all through these Zoom meetings. So I hope we'll get the Pathways group well represented at the scientific sessions. And I want to announce that there is a Pathways Symposium in the planning stages for Friday, June 3rd. And again, the particular format and the goals of that session is something that we want to bring forward in our discussion on February 2nd. So 3 sessions planned here for early 2022. The one on February 2nd will be Mike Stitzel with a scientific presentation, and then a discussion of future pathways activities, and a regular scientific section on March 2nd. So I hope everyone will plan to come to those sessions. Put a hold on your calendars for the second Wednesday of the month in April and May, and we'll keep things rolling. So with no further ado, let me jump right in and introduce our first speaker today, Ebony Carter from Washington University in St. Louis. Ebony received an Accelerator Award in 2019, and she's going to tell us about targeted lifestyle change groups in prenatal care. Is this a pathway to stop diabetes? Ebony, please.
Thank you so much. Good afternoon, everyone. Happy New Year. And thank you so much for providing this opportunity for me to share our work. I have no financial disclosures. And in terms of an outline, I'm going to give you some background in terms of how we got to this project and then some of the other work that we've been doing. So any of you who were here when I first got the award have seen this slide before. I created it for my Pathway finalist PowerPoint, and I have used it in almost every presentation since, despite the fact that my mentor had hated it and was like, it's too busy. But I love it because it grounds me in terms of the work that we do. And so if you think about pregnancy over a life course perspective, we're born, you're in pregnancy, you take a little bit of a metabolic hit, but you're young, you're healthy, you go back to normal, and you live your life. But a lot of patients, and the ones that I serve, live on the yellow line. And so they are at higher risk. Let's say that they're born to a mom that has gestational diabetes. And so they take that hit and they cross the threshold into GDM when they're pregnant themselves. They're young, they're healthy, they go back to normal. And they're at higher risk for getting type 2 diabetes later in life. So the goal of my research project and everything that I do is to essentially push patients from this yellow line down to this green line so that hopefully they don't get gestational diabetes or type 2 diabetes, or if they do, that the effects are not as severe or that happens much later in their lives. So one of the major ways that I have tried to do this is through group prenatal care. And just so that everybody's with me, because you may have never heard of it before, group prenatal care is a fun, innovative form of care. It was started in the 90s by a midwife for low risk patients. And essentially just picture that you yourself or a loved one is pregnant and in the waiting room, you go in to get your prenatal care. And rather than sitting in the waiting room for 30 minutes waiting for your doctor or midwife, you go right in, you get your snacks, you check your own blood pressure, you check your own weight. Everybody has a brief assessment period. That's what you're seeing. This is actually in the room where we do our group care. So you meet with the physician or the midwife, check the baby's heart, make sure everything is going okay, ask any questions you have. And if it's a question that other patients in the group probably have too, then I'll say, you know what, I bet somebody else has that issue too. Do you mind if we table that for the group? And they usually say yes. And so as a clinician, you're not saying the same thing a billion times a day. So you do that brief assessment, and then you've usually done all of the patients within 30 minutes or so. So the next 90 minutes are spent with 6 to 10 other patients of the same gestational age doing fun, interactive learning activities. So in a typical 10-minute visit, you don't have time to talk about depression and infant care and how do you know that your baby is hungry and all of these things, but you have time for that with this 2-hour every other week prenatal care situation where you get all of your care, one-stop shopping in the same place. So this was originally done for low-risk women. I'm a high-risk obstetrician. And so I wanted to see, are some of the tenets that have worked really well for low-risk patients true for patients with diabetes as well? In the low-risk literature, there's significantly lower rates of preterm birth and low birth weight and retention in care and patient satisfaction. And that's also true outside of pregnancy. It's usually called shared medical appointments, but there's also really good data that group care interventions have improved glycemic control for patients with diabetes. So this is just one of several meta-analyses that have been done showing a 0.5 percentage point decrease for patients who were randomized to shared medical appointment interventions who had diabetes. And I hope that everybody is familiar with the forest plot for meta-analyses. If you're not, if you could just put a hand up or something and I'll go through it quickly, just because I'm going to show a few of these throughout the talk. And seeing no hands, I'll keep going and assume that everybody is with me and knows how to read a forest plot. So we started the diabetes group care program at Washington University in St. Louis. When I was a fellow, I didn't know what I was doing. And this is a picture of the 6 patients who went through it with me. And I basically said, I don't know if this is going to work, but I think it will improve outcomes. This is a picture of a birthday party that we had a couple of years later with that first group. And this is us trying to do yoga. So with that pilot curriculum that we did, we actually did a pilot randomized study in 80 patients who were randomized 1-to-1 to either traditional care in our Center for Diabetes and Pregnancy or to this diabetes group care intervention. I'm just going to share with you a few highlights that we found from that trial. So patients were randomized at 22 to 34 weeks gestation because I originally designed it for patients with gestational diabetes. But there's one problem. I practice at a tertiary referral center in the middle of St. Louis, and the majority of our patients already had type 2 diabetes. So I didn't have enough patients for a GDM trial. So out of really necessity to keep the groups large enough to work and to be cost effective, we ended up including patients with both GDM and type 2 diabetes. So since we didn't start until 22 weeks, gestational weight gain overall, there was no difference between group care or individual care. Then we looked at, did they achieve the IOM, or now National Academy of Medicine gestational weight gain recommendations? Once again, there was no difference. But since they didn't start the trial until 22 weeks at the earliest, we actually looked, once they entered the study, week by week, what was the gestational weight gain, and saw that patients who were randomized to group actually had lower rates of gestational weight gain once they were in the study. But by this point, they were halfway through their pregnancy. Now, if we just look at the patients who had type 2 diabetes and their glycemic control, you can see that the patients who were in group, their starting A1C, this is higher than we want. We want patients in pregnancy to have an A1C less than 6%. And then we looked at the final A1C prior to delivery, and they're not significantly different, but these are tiny numbers, and it suggests there might be a trend there. So we wanted to look into that further. And then finally, and I feel like this is where the money is, for patients just with GDM who were in this study. Patients in pregnancy, it's really hard to get our patients to come back for a 6-week postpartum visit. And the rates of having a 6-week CT for if you have GDM, it's really only like 20% of our patients are actually coming back. So we don't know whether they have type 2 diabetes or not, because we don't see them again once they deliver. But for patients randomized to group, 70% of them came back. So almost 3.5 times more likely to come back for your 6-week glucose tolerance test if you were in group. And I would suggest that that's because they saw patients with type 2 diabetes. They understood what they were trying to prevent and what needed to be done. So, in pictorial form, I kind of have my pregnancy study, with the gestational diabetes and the type 2 diabetes. We actually have a definitive NIH funded trial of that's actually scaled to see, are there actual differences in glycemic control? We're following those moms and babies out for 1 year postpartum to see, do the effects of the intervention actually endure? And then ADA and the ADA grant really came about because I felt like we were doing too little too late. Like the horse is already out the barn by 22 to 24 weeks when we're doing this intervention. And so what if we pulled it back into the first trimester and did a lifestyle intervention to see if we could provocatively stop diabetes? And so that's how TLC was born. It stands for Targeted Lifestyle Change Group Care. And we kind of have several components of it. So for every session, this is the curriculum. We worked with a multidisciplinary team. We did a literature review, so saw what other people were doing, what we had done with our diabetes trials, and had me, a midwife, a nutritionist, a diabetes educator, former patients who have been in our group care program, and developed this curriculum. Every single session hits a pregnancy topic, a nutrition, an exercise, and a behavioral health topic. And we don't hit all of this. I mean, it's too much. The idea is that you go where the patients needed to go. So if I was doing a group today, we'd probably be talking about Omicron. You do what's important to the patients at that time, and they really are guiding what happens, even though we do kind of have a set notion of what we might cover during that session. So with TLC, we're randomizing 416 patients who have a BMI greater than 25 and greater than or equal to 1 GDM risk factor. And just so you know, that's our entire patient population, right? GDM in a prior pregnancy, basically a high risk race, which is essentially anyone who's not white, physical inactivity, PCOS, it's our entire patient population. So we're randomizing them 1-to-1 to traditional group care or TLC, and then we're following them out to see what happens with the mom and with the babies. So we started randomizing patients back in November of 2019, and the y-axis is the number of patients, the x-axis is time, and so you see November all the way back up to December of 2021. And so the bars are how many patients we randomized in each of these months. And so it was actually going really well. By March of 2020, we essentially had 40 patients randomized to the trial. The groups were going great, patients were enjoying it. And then you can see what happens with this huge, dive off of the cliff, and this is the COVID pandemic. So all clinical research operations at our institution were halted. All group care interventions, even just the medical ones, were halted for a year. So luckily, in the summer of 2021, we were able to resume recruitment, and it actually started going really well again. And now we come to January of 2022, and as of Monday, all group care interventions at our institution are again shut down. So essentially, whenever we're able to actually recruit and do the trial, it's going extremely well, and the patients are excited about it, and the pandemic. So, what do you do when a pandemic tries to block your pathway grant? What I have done, and I'm going to share with you some of the other things that we're working on now, are meta-analyses. Because in addition to my TLC trial with the pathway program, also the diabetes trial that we're doing is also on hold. The diabetes trial essentially is exactly the same thing we did with the pilot, just now scaled. And so I have, this population of patients that are kind of on hold, and it's going to be a few years until we get those results. And so we essentially did a meta-analysis with all of the other studies, around the globe that have done diabetes group care interventions. And so the question is, what is the effect of group prenatal care on preterm birth rates among pregnant patients with diabetes compared to traditional care with a lot of the other diabetes outcomes as well? So this is actually unpublished data. I'm working on it now. There's still one trial that didn't have enough information in the original paper for the meta-analysis, and the authors are sending me those data now. So this is not finished, but in terms of what we have so far, this forest plot graphs the randomized trials together. This is my the pilot that's done. These are just a few patients of the one that is the follow-up study. And then these are the observational studies. And you see that there's some suggestion that patients who are randomized to diabetes group care probably have lower rates of preterm birth. We also looked at large for gestational age, and there was actually, in the meta-analysis so far, no difference. And we'll have another 500 patients coming from the trial that we're waiting for. And then we looked at NICU admissions for the babies, no difference between diabetes group care and traditional care. And I realize I'm going through these fast. So I'm going to slow down on this one because this is back to me, the money slide. So for patients who had GDM who were randomized to diabetes or who were in diabetes group care trials, you can see that there's no difference for the RCTs. And these are really just the one that we finished and the one that we're working on now. One more is coming. But for the observational studies, there was a difference in the rates of patients coming back for their 6-week GTT. So overall, it looks like there probably is a trend towards patients being more likely to come back for GTTs. And I keep harping on this. Non-obstetricians probably don't appreciate this fact, but people have done all kinds of things to get patients to come back. Telephone visits and, navigators and all, nothing has worked. So this is really the largest effect size that I've seen in the literature. Oh, and that's just showing that 77% with group care and 55% in individual care came back for that. So what else do you do when a pandemic tries to block your pathway award? Editorials. So this is an editorial from JAMA that I wrote with Camille Poe, talking about like the increasing rates of gestational diabetes. And then this study also kind of parsed it out by race. And we also wrote an editorial for the Green Journal, the Journal of Obstetrics and Gynecology, about screening strategies. And so in pregnancy, there is still tons of controversy about how you diagnose GDM. Do you use the one-step test that the rest of the world uses, which is like your standard 2-hour, or do you use a 2-step screening, with a threshold of 130, 135, or 140? And if you fail that, then you take a 3-hour test. So tons of controversy. Everybody does it a little bit differently. So it's very difficult for the GDM literature to parse out what's actually happening. And so 2 large randomized trials were done in 2021 that actually looked at large populations of patients and, does it matter whether you do a 1-step or a 2-step? One of the issues with the 1-step is it diagnoses a lot more patients with gestational diabetes, but at what cost? So you're going to have more patients who are getting the diagnosis. You're going to probably treat them more aggressively, but does that actually benefit mom? Does that benefit baby? And so both of these trials came out and suggested that they don't. So as we expected, they saw much higher rates of GDM, but there was no commensurate improvement in maternal or neonatal outcomes. And what Camille and I argued in that editorial is that if you only care about pregnancy, then it seems like the 2-step test is probably winning, but that's short-sighted. I told you I care about a life course approach. And so if you're looking at it from a life course, if a patient is diagnosed with GDM and gets that intensive lifestyle management, does that change what's happening to her 15 to 20 years? And those are questions that were not answered by these trials. And the other thing is, were they adequately powered for the neonatal and the infant outcomes and what happens to that baby and then that baby throughout childhood? So this is more unpublished data. But all of the trials are here. So we're about ready to submit it. So this is now looking at for GDM diagnosis, if you have the 1-step test trials versus the 2-step. Once again, I have the randomized trials at the top, the observational trials here, and then our pooled effect sizes down here. And so as expected, for patients who had the 1-step test, almost 10% are going to be diagnosed with GDM versus for a 2-step test, it's only going to be like closer to 4%. So that's pretty standard. Back when they published the HAPO study in 2008, that was kind of a known phenomenon. Now, if we look at large for gestational age, and I have it parsed out by randomized trials, observational, or all, and then these are the percentages with the 1-step versus the 2-step. So just with the RCTs, there's no difference in LGA. But when you look at the observational trials, it looks like there is a significant difference. And then the observational trials are driving this, but maybe a modest decrease in rates of LGA for the one-step testing. Remember, these are people who are more likely to be diagnosed and then in turn more likely to be treated. I'm going to skip the funnel plot on this for the sake of time. And then if we look at the maternal outcomes, so if you're diagnosing all these additional moms, are you actually improving their outcomes? And interesting, it looks like probably not. So it's not surprising that more of them are being treated, but more of them are getting C-sections. There's no difference in hypertensive disorders of pregnancy. And then if we look at the baby, this kind of follows the large for gestational age. And I should just say large for gestational age is based on birth weight controlled for gestational age of delivery versus macrosomia is just 4,000 grams. So whether you were born with 4,000 grams at 42 weeks or 37 weeks, it's going to count here because it doesn't account for gestational age of delivery. And so lower rates of macrosomia for the one-step testing, but neonatal respiratory distress syndrome was higher for the one-step. Neonatal hypoglycemia. So it almost makes you wonder, we're diagnosing all of these additional people, and maybe they're getting a modest improvement with regard to their large for gestational age, questionable because the RCTs didn't show it, but we're doing more stuff to them. They're more likely to get C-sections. Their babies are more likely to be hypoglycemic. So it kind of makes you wonder, are we almost over-treating in that situation? We make the argument that in terms of neonatal outcomes, I would argue that the modest improvement for LGA probably is not offset by these other things that we seem to be doing to the patients, but I'm sure that this debate will rage on, and this will be our small contribution to it. I have a couple of minutes left, and so I will close with one final study. I told you that we practice in St. Louis. I serve a patient population that is 80% Black, predominantly insured by Medicaid, and so we notice that patients come to us, we're not in a Medicaid expansion state, so patients come to us with their diabetes completely out of control because they've often had no care. So it's not unusual for me to see an initial OB visit and the patient's A1C is 12 or 13. And so, is prenatal care enough to overcome all of that accumulated lifetime of stuff? And then we get them for 9 months, we do aggressive control, and then what happens? Does it overcome what happened before? So we wanted to determine the impact of neighborhood level factors on pregnancy outcomes in women with type 2 diabetes utilizing the area deprivation index. And I have a list of the things that go into the area deprivation index here, but it goes beyond just kind of like income at a census level track, because you're getting all of these other things like home values, income disparities, owner occupied, families below the poverty level. So all of these additional factors go into calculating an ADI. And they're done both for states, and then there are also national levels so that you can do it, either way. And this is just kind of a heat map of the ADIs for the census tracts in St. Louis. So to orient you, this is the Mississippi River. This is downtown St. Louis. This is the star where I am sitting right now at Barnes Jewish Hospital. And red essentially means high ADI or high deprivation, and blue means low levels of deprivation. So you can see in the western suburbs, where the preterm birth rate looks like Western Europe, there's not a lot of deprivation. But if you go north of St. Louis, and if you go east of the river over here in East St. Louis, highly deprived neighborhoods, and this is where my patients tend to come from. And so we classified our patients as low versus high ADI, and I'm gonna skip forward to this slide. So these are the national percentiles of ADI. And essentially you can see that our patient population is highly deprived. And so really what we're comparing is high deprived versus moderately deprived. And there are differences there. So in the highly deprived neighborhoods, they were more likely to be Black. The diabetes severity of disease tended to be higher if you were in a high ADI group. And I already showed you the distribution. I have 1 minute left before I open up to questions. So I'm gonna just show you this because I think it visually shows what I wanted to say. So we looked at patients' A1C levels in the first, second, and third trimester. And these are patients who had type 2 diabetes in pregnancy. And you can see that all of them are coming into pregnancy with high A1Cs. So, I have it in the next slide. So, almost 9 and then almost 8 for the less deprived neighborhoods. And then you follow them over the course of pregnancy, and you know that our management is very aggressive. And so by the second trimester, we have all of them essentially less than 7. But if you follow them into the third trimester, and this isn't statistically significant, probably because of our sample size, but there's still a disparity there. So prenatal care is great in that we do a lot to bring patients under good control. But you know, it's still not perfect. And you can also see that our goal is that patients are going to be less than this 6%, assuming that they're not having lows. You can liberalize that if it's low, but our patients are a lot better, but still not less than 6% at the end. And these are, I can go into this more with questions, but these are just kind of diving into that data a little bit more deeply. So in terms of acknowledgements, I would like to thank the Pathway Grant. I see Barbara, my wonderful mentor. It's so good to see you on today. She's been an amazing help. So thank you so much for being here. And I also just wanted to say that Pathway has opened up so many opportunities for my career. When I look at like the last 2 years of having this grant, 2 or 3 weeks after I started, my third daughter was born. So that's a Kinsey of hope. I received my first R01. You know, the pandemic has been rough, but some good things happened during it, including my first R01. Was named chief of our division of clinical research for OBGYN here at WashU. Got promoted to associate professor last summer, earned tenure in October. And then, oh, I'm sorry, I forgot I had things to go with these. And then the Green Journal is our journal for the American College of OBGYN and was named their inaugural editor for equity. Associate editor, I should say, last month. And so I just wanted to say thank you so much to the Pathway program, because the Pathway opportunity really has catalyzed all of these opportunities. And I look forward to Omicron getting better so that we can restart TLC and get this trial back on the road. I'd be remiss because, you know, research is a team sport. And so I am eternally grateful for my research team who work tirelessly, and I am so grateful for them. And with that, I will see if there are any questions. And I couldn't have the baby in and not put the other two. So these are my 3 babies. I'll open it for questions now. Thank you.
Thank you, Ebony. That was terrific. Let's see, we've got a question in the chat from Ernesto Mizrachi. I enjoyed your presentation. I was wondering how the interventions during pregnancy align or complement the DPP, the diabetes prevention program after delivery, and how is this population followed long-term?
That's an excellent question. And my answer to that question is actually fairly disappointing. So I mentioned that we are in a state that did not expand Medicaid. And so most of our patients lose their insurance 45 days after delivery. And honestly, that first group of patients that I showed you, it actually, when they got to their 6-week visit, and it was kind of like we saw the babies come back and everybody's thrilled, I didn't have a good bridge to care for them because they had lost insurance at that point. And so it almost feels unethical. You have patients who are activated, engaged, and know exactly what they need to do, and then don't have the care they need. And so there's a community health center that a lot of our patients then go to. But essentially the answer is there aren't the resources that are available to continue to support these patients in the way that they really deserve to be supported. So in the diabetes trial, this next one, we're following them for 1 year postpartum, but there's not actually a formal program in place like the DPP for those patients at that point.
Okay, I see Andy Stewart has his electronic hand raised. Andy?
Yeah, thanks, Ebony. That was really a great talk, and I can certainly understand how difficult this stuff is. It's next to impossible. But here's my question. I'm wondering about the impact of CGM on all of these outcomes. And, you know, obviously it's a population that probably doesn't even have access to any CGM, but what is the world's literature on CGM in GDM and preventing, bad outcomes down the road? Is there a literature on that? And
so, I mean, essentially for GDM, our patients don't have CGM, like they don't have access to it. So there are studies. I know that Camille at MGH has like studies where they have kind of like, you can check out CGMs for patients that are hard to manage, but it is not part of the standard of care. I mean, to be totally honest, I have trouble getting CGM for my patients with Medicaid who have type 1. So, you know,
I wonder if Medtronic might like to sponsor such a trial for you.
Yeah, well, and I know that there is and these patients don't even have diabetes yet, but there's an NIH trial now through NIDDK where they're using CGMs for patients who I think are at high risk of getting GDM and following them forward. But that's all I'm really aware of.
Yeah.
So,
thanks. It's tough.
So Ebony, I was just wondering, the salvage of the cohorts as you have hit the cliff of the two big COVID events, the one that's happening now and the one that, came before. It seems like in order to have a complete data set, your mothers have to go through to childbirth. And I bet that many of those pregnancies were interrupted in a sense by the, shutdown, the COVID shutdown. How do you salvage, some of the work that you've done, and how much can you still pull out if you haven't been able to take the mothers all the way to term?
Yeah, I mean, because honestly, my hope was, I have to regroup now because Monday kind of hit us by storm because we just got shut down again. But I actually had planned to like over recruit to get us back to the right number of patients to be powered appropriately, because those first 40 patients essentially only got 2 or 3 groups of the session before it was gone. We also experimented with going to a virtual platform, not just for TLC, but for our groups overall, just so that we wouldn't lose so much time. And what we found is that groups that were already established did okay with a virtual platform. If they weren't established, people just didn't come. It's an hour or 2 of being on, people are just zoomed out. There were problems with like the technology and having access and connectivity. And so we tried the virtual platform, and there's an X factor to being with other pregnant patients in a room and the social support that happens with it that we just could not recreate on an online platform, which now I really wish we had, now that I find myself in this predicament yet again.
Yeah, but still a valiant effort, and really, like Andy said, really nice presentation. Thank you so much.
Thank you.
I think we're going to move on to our second talk, which will be by Jonathan Flak. Jonathan, if you want to get your slides up, that would be great. Jonathan comes from the Indiana University School of Medicine. His award was in the initiator category in 2017. And the title of his talk is Neural Pathways that Restore Energy Balance and Euglycemia. Jonathan.
Thank you for that introduction. And thank you all for the opportunity to present today. And I also wanted to thank the organizing staff for organizing this seminar series that for me has been a real treat throughout the COVID-19 pandemic. And for today, as you stated, I am a ADA Pathway Initiator awardee. And as such, I'm going to begin with some work that I was able to conduct with Martin Myers at the University of Michigan, and then transition into my own independent work that I've been able to accomplish in Indianapolis. So as the focus of my Pathway project, I'm defining the underlying mechanisms for initiating the key responses that protect normal individuals from deadly hypoglycemia. But these responses can become insufficient to protect diabetic patients and be a barrier for treatment. And insulin-based therapies carry with them the inherent risk of developing hypoglycemia in diabetic patients. And if blood glucose levels fall low enough, they can induce diabetic seizures and diabetic shock, which require emergency treatment. And if these patients don't receive the required treatment, there can be fatal consequences. And one of the main factors that contributes to the susceptibility of hypoglycemia in diabetic patients is the onset of hypoglycemia-associated autonomic failure, or HAFE for short. And this is where recurrent episodes of hypoglycemia suppress the responses to future hypoglycemic events, making the individual more and more susceptible to hypoglycemia over time. And the primary protection from deadly hypoglycemia is the counterregulatory response to hypoglycemia. When blood glucose levels fall low enough, cells in the brain become activated, which ignites a neural circuit that triggers the coordinated release of glucocorticoids, glucagon, and catecholamines. And in hypoglycemia-associated autonomic failure, or HAFE for short, the ability to release catecholamines during hypoglycemia is no longer sufficient for mobilizing sufficient amounts of glucose within the circulation, which in turn makes the individual increasingly susceptible to hypoglycemia. Some details are known about the underlying neural mechanisms for the generation of the counterregulatory response and how these mechanisms become dysfunctional in HAFE. For example, the role of the ventral medial hypothalamic nucleus, or VMH. But many of the details, including efferent nodes for how these signals trigger the release of these hormones, is not well understood at this time. So my project was aimed to better identify the cells in the VMH that generate this response and to uncover the details of this neural circuit by which it acts on. So this goal seems easy enough until you realize a problem with the brain. Each area doesn't necessarily perform an individual function, and more commonly, there are intermingled cells within a given area that sometimes perform opposing functions. And such is the case with the VMH. Intermingled within the nucleus are cells that have the potential to stimulate opposing aspects of glucoregulation. They can stimulate glucose transport, and separate cells can stimulate the mobilization of blood glucose from energy reserves. So the presumably ultimate function is to tune the function of separate organs, and these individual cells then contain unique receptors and neuropeptides that hopefully can be leveraged within future. When we started this project, we previously studied a set of cells in the parabrachial nucleus that project into the ventromedial hypothalamic nucleus. These cells are inhibited by leptin. They release the neuropeptide cholecystokinin to mobilize glucose when blood glucose levels fall. So you then presume that the VMH cells would then contain the cholecystokinin receptor. And indeed, there are reports that the VMH contains dense expression of the cholecystokinin receptor B. And this receptor is known to stimulate the sympathetic nervous system activity, which is a key component of the counterregulatory response. So we generated a CCKB receptor Cre knock-in mouse line that reveals Cre activity in the VMH in a GFP reporter mouse. So it's great. It means that the Cre works. And we can actually use this Cre activity in order to study these cells. And one thing we were able to find is when we treat these animals with leptin, leptin induces phospho-STAT3 in virtually no cells within the dorsal medial division of the VMH that contain these green cytosolic markers, meaning that these CCKB receptor neurons are separate from those that are responsive to leptin. So now what happens when we activate these CCKB receptor neurons within the VMH? Well, you get an even greater hyperglycemic response than activating all the VMH togethers using SF1 Cre. And which we think that this is due to the fact that we are primarily activating these VMH cells that drive the counterregulatory response and not the ones that can stimulate glucose clearance. So as I previously stated, we're not only just interested in understanding the generation of the counterregulatory response, but we're also interested in the onset of hypoglycemia-associated autonomic failure. And we have a model that we study in the lab that we've been successful with, that if you treat animals with consecutive days of 2-deoxyglucose over the course of 4 days, by 4 days that this response gets blunted over time. So what we are interested in is this ability for VMH CCKB receptor neurons to push up blood glucose, is this blunted by a history of hypoglycemic or hypoglycemic-like events? And the answer is yes. So here in black is an animal that was receiving vehicle treatment, activate these VMH CCKB receptor neurons with the light, and there's a potent mobilization of blood glucose that's suppressed if there was a history of glucoprivation. So overall, these results tell us that CCKBR VMH neurons can mobilize blood glucose, and that function is suppressed following a history of hypoglycemic-like events. So in addition to understanding the potential function of activating VMH CCKB receptor neurons, we're also interested in what happens when we take them away. So we elected to use a tetanus toxin means in order to prevent vesicular communication from these cells. And if we chronically prevent their ability to function by vesicular means, what we found was that silencing these VMH CCKB receptor neurons lowered blood glucose at both a fed and a fasted state. Now, the question is, are these mice still able to mount this full counterregulatory response? And the answer is no. So in response to either insulin or 2-deoxyglucose, in either case, there's a suppression in the glycemic response to either stimuli, which is consistent with a suppressed counterregulatory response to hypoglycemia. And in examining these mice more closely, what we're able to observe was that in these mice, at the same time as they have these lowered basal blood glucose levels is they also have elevated glucagon and reduced insulin, which you may be sitting there thinking that, well, that doesn't make sense because, of course, if they have lower blood glucose, they should have higher insulin and lower glucagon. And I agree with you, but I think what is actually happening is that the pancreas is actually counteracting the effects of the brain that's happening on suppressing these responses. And the pancreas is protecting blood glucose levels such that these blood glucose levels would be even lower if these responses were not happening at the level of the pancreas. And in support of that notion, what we did is we treated animals with STZ to induce beta cell ablation. And what happened is blood glucose levels did not rise as much as control animals, indicating that silencing VMH CCK-BR neurons have the potential to lower blood glucose at baseline in response to hypoglycemia and also in response to beta cell loss. However, despite these changes in blood glucose, there is absolutely no effect on energy balance in these mice, such that as we measured body weight, food intake, VO2, fat mass, and activity were all completely normal in these mice. But we were able to observe the fact that if you silence all VMH cells using the SF1 Cree tactic, that there was obesity set into these mice. That obesity was not associated with an increase in food intake. It was associated with a suppression of VO2, increase of fat mass, but no change in activity. So this indicates that there must be non-CCKB receptor neurons in the VMH separate from these cells that generate the counterregulatory response to hypoglycemia that are essential for energy balance through the stimulation of energy expenditure. So as we update our model, we have two separate sets of cells within the VMH that perform two separate metabolic functions. One acts likely through separate peripheral organs in order to stimulate energy expenditure, whereas the CCKB receptor neurons project to potentially other organs, such as the adrenal and the liver, to ultimately mobilize glucose and glucocorticoids in order to promote the euglycemia in the face of hypoglycemia. Now, Greg Morton and Mike Schwartz were able to reveal that the output from the CCKBR neurons was the anterior BSD. However, the central output from the VMH that performs the function of initiating or stimulating energy expenditure has not yet revealed. So in order to understand these cells a little bit more closely, we need a way also to tap into these cells to understand the function of these VMH cells independently from the CCKBR cells. And we thought, well, possibly we could use the fact that Simon Luckman previously reported that the expression of the neuropeptide pituitary adenylate cyclase activating polypeptide, or PACAP for short, is suppressed by either fasting or a loss of leptin action. So potentially that's a mechanism in which the system is suppressing energy expenditure in response to periods of negative energy balance. And so we tested, and then we would expect, if that's the case, we expect that if we lost PACAP action within these VMH cells, we should induce obesity. And so we tested whether the loss of PACAP within the VMH would lead to suppressed energy expenditure and obesity. So in order to accomplish that, we acquired a mouse from Brad Lowell in which the gene that encodes for PACAP, ADCYAP1, was Flak's. And because SF1 can be expressed in the periphery where PACAP can also be found, we elected to eliminate PACAP in the VMH by an AAV Cree injection locally in the VMH. So using these AAV injections, we're able to almost completely eliminate the expression of ADCYAP1 in the VMH using in situ hybridization, but not affect other structures throughout the hypothalamus that are also contain dense clusters of PACAP. However, it's important to note that these AAV injections were not, we have absolutely no evidence of the fact that we've ablated the nuclei. And in fact, if we look at either NUN or DAPI staining, that these cells look like they're completely present and they're active even in weeks. This is 12 weeks following the administration of the AAV Cree, they still look like they're present. So now what happens to the animal once we eliminate this pay cap within the VMH. Well, the animals become obese, and similar to chronic silencing of the VMH SF1 neurons, there's only a hint of an increase in food intake, but no effect on a fasting refeeding experiment, which suggests that this increase in body weight is likely due independently of food intake. Now, I can't rule out that there's absolutely no role of this increase in food intake, but at this point, the animals are already 30 to 40% larger than their GFP littermate counterparts, that likely food intake is not the main driver of this effect. Now, when we go to the metabolic cages, and this is at 4 weeks, so 4 weeks is when the animals are just starting to tick up, where there's there's starting to be an increase in body weight. And what happens when we look at the metabolic cages, indirect calorimetry in those mice, what happens is VO2, especially during the dark, is suppressed, as well as VCO2. But again, no difference in activity. So I had a collaborator who suggested that we really need to focus on body temperature, given the connection of the VMH with brown adipose tissue function. So we were advised to look closer into whether core body temperature was changed in these mice. And I've tried multiple cohorts, and using a rectal probe thermometer, had absolutely no hint of an effect of core body temperature in these mice. So at this point, I thought that there was absolutely no effect on body temperature, and I should move in a different direction. However, I had a collaborator at Indianapolis who was very insistent that I need to move to a different way in order to measure body temperature, and more locally into different adipose tissue depots instead of just globally body temperature. So what we started doing in the lab when I moved to Indianapolis was we started to use these temperature sensing microchips into both white and brown fat. And so we chose to compare the inguinal fat as our beige depot and use the interscapular brown adipose tissue as our brown tissue. And we eventually were able to reliably embed these temperature sensing microchips into the fat pad and be able to accomplish this in the same mice. So within all these same knockout mice, we're able to measure both inguinal fat temperature as well as brown fat temperature, all in the same mice. So we started with a simple experiment to see, well, is there a drop in temperature above the brown adipose tissue that follows what happens with the rise in body weight in these mice? And the answer is yes. So when we look at, especially within the brown fat, is that pretty soon after the AAV pre-injections is that the temperature begins to drop. That's significantly different along the experiment. And this only happens in the brown fat and doesn't happen in the white fat. But in addition to this, we were very interested in what happens not only at this point, which is midday lights on. So this is a point where the animals have particularly low metabolic activity. They don't really move around much, and they're pretty inactive. But what happens when animals are feeding? What happens and so what we began to do was to look at what happens after the lights off. So after lights off, mice are nocturnal and they begin to eat. So if you look at the control animals just before lights off, and just at lights off, there's a rise in their IBAT and IWAT temperature that rises over the course of 2 hours after lights off. And then when we look at the knockouts, this effect is suppressed. So this was encouraging that we need to be focused more on the behavior of these adipose tissue depots in these knockout mice. That's potentially explaining the obesity effects that we're seeing. So, but certainly this is also going to have a circadian effect. So in order to look at responses to feeding in a slightly different way, we then aim to drive their feeding response at this period of low metabolic activity during midday lights on. So in order to do that, we fasted animals for 24 hours. And then when we, oh, there we go. Okay, so when so this is animals pre-fast, and then after post-fast, bat temperature and IWAT temperature drops, which then when we return the animal's food, all of a sudden temperature increases at both above the IBAT and the IWAT, such that when in the knockout mice, this effect is suppressed. So this is making us think that really what we're looking at is some connections that come from the VMH that eventually descend on brown and beige fat in order to stimulate metabolic activity at these tissues that is no longer functional due to the absence of this neuropeptide. So what we think is that there are that in the VMH on these PACAP containing cells that they sense overall signals that regulate the diet, such as leptin, T3, and glucose. And then these cells signal to a cell downstream in order to increase VO2, VCO2, IBAT temperature, IWAT temperature, mitochondrial coupling, which together what they do is they burn excess energy when needed. And the way this works is during negative energy balance, such as weight loss or fasting conditions, these cells no longer get that signal, and they no longer drive these responses, which leads to the restoration of energy stores. However, when you eliminate PACAP, so again, you have the presence of leptin, T3, and glucose, but they're no longer in order to stimulate this response. What happens is in the context of this situation, there's excessive energy storage. And this occurs presumably if you silence, ablate the VMH, then there's the same buildup of energy storage because of the absence of this response. But the question that we had is that, well, how are these VMH signals signaling downstream? Where is this cell? What is this cell that's performing this individual function? And can it be used in order to override and restore normal function in an animal with VMH ablation? So in order to answer that question, what we turned to was anterograde tracers. So in this case, we used a Cre-inducible AAV synaptophysin M cherry that's enriched in the synaptic terminals. So we injected this into the dorsal medial VMH and found that there were projections into the preoptic area, the bed nucleus stria terminalis, periventricular thalamus, and periaqueduct gray. Again, no surprises in those areas. But the one that hasn't been studied to nearly the same degree was the dense projections that we're able to observe in the preoptic area. And so this is particularly in the caudal regions of the preoptic area that lie just lateral to the suprachiasmatic nucleus and extend a little into the same slice as the bed nucleus stria terminalis. And given the role of the preoptic area in thermoregulation, we hypothesized that there must be cells that the VMH must contact that are necessary for body temperature control that can stimulate thermogenesis in adipose tissue and muscle that is critical for energy expenditure stimulation. And if this is the case, then these cells must be able to be stimulated to induce weight loss and are necessary for energy balance and responsive to changes in energy states such as fasting refeeding. And what we're able to observe is that when we used an SF1 dread mouse, so this is using the activating form of the dread, activate SF1 neurons within the VMH, what we're able to observe is that there's an increase in CFOS within this area of the preoptic area that we see these projections. And this occurs in both males and females. And in examining sections from a different experiment, we're able to observe that there is a complete absence or almost large absence of CFOS positive nuclei in fasted animals compared to fed animals. So we ran a small experiment and we saw that refeeding actually increased the number of CFOS positive nuclei within this division of the preoptic area that we see these VMH positive fibers. So we next wanted to find, well, how do we figure out what are these cells in here that are potentially driving energy expenditure that are responding to the VMH? Well, one benefit is that the single cell sequencing from the preoptic region has already been published. And this is from a group at the Broad. However, it's important to note that this dissection does extend far outside the area that we're primarily interested in. And they do define this as the preoptic region and not necessarily the preoptic area. But using this approach, they were able to define 87 clusters of cells, many of them neurons. In order to make it a little bit easier to digest, within the paper, they separated out inhibitory, those that contain GABA, versus excitatory cells, those that contain glutamate. And we decided that maybe we should take the same kind of approach in order to at least break the options in half in order to make it a little bit less overwhelming needle in the haystack approach. So going back to our model, it indicates that these cells that drive energy expenditure from the VMH contain the neuropeptide PACAP, and presumably we hypothesized that they contact this area, the preoptic area. So what we can do is to test whether these cells do have the potential to stimulate energy expenditure by comparing the ability to activate them using HM3DQ, CNO administration to activate HM3DQ in either VGAT Cree mice or VGLUT2 Cree mice, or compare the loss of function approach by eliminating either VGAT or VGLUT2. And what we saw was that if we activate either VGAT or VGLUT2 cells within this area of the preoptic area in which the VMH neurons project to, what we see is that if we activate these cells for 3 consecutive days, we get between a 1.5 gram and a 2 gram loss of body weight, which unfortunately that doesn't really help answer my question, because in both cases, either cells are decreasing body weight to a similar degree. However, when we looked at food intake, only activating the VGLUT2 cells in the POA led to a suppression in food intake. So this point in this direction that we should be focused on GABAergic cells. And I mentioned earlier that the function of this area is primarily in thermal regulation. So there must be cells within the preoptic area that suppress body temperature. So we want to make sure that these animals weren't leading to a large decrease in core body temperature. However, it looks like, at least in this case, that by activating the glutamatergic cells, do we have this acute drop in temperature that occurs within these mice. So in further support of the notion that we should be focused on GABAergic preoptic area cells that promote energy expenditure, we then eliminated either glutamate or GABA within this area, and only within the VGAT knockout mice did they have an increase in body mass and fat mass. So for our model, we now think that these PACAP cells project into the preoptic area on GABAergic cells in order to promote the burning of excess energy. So, but obviously we want to get closer, or we want to get more specific than just eliminating GABA versus glutamate. So one way in order to get closer to identifying that needle in the haystack is to use single cell data sets in conjunction with bulk RNA-seq. And so we are collaborating with Travis Johnson at IUSM in biostatistics, and he has bioinformatics and machine learning capabilities set up at the Institute to perform enrichment analysis in order to define whether the defining transcripts of each cluster are upregulated, downregulated, or a combination of the two. His background is in Alzheimer's disease and cancer, but we have gotten him interested in diabetes. So what we did is we took either mice who are fasted for 24 hours or on 16 weeks in a high-fat diet, pulled out their preoptic regions, similar to what they used from the group at the Broad, and did bulk RNA-seq. So now we're looking at some group of cells that is potentially downregulated by fasting and upregulated by a high-fat diet. And for each condition, either fasting or high-fat diet.
I'm sorry, we're going to need to wrap up here.
Okay.
Time. Thank you.
Fine. No problem. All right. Well, thank you for your time. I will take any questions.
Sorry, I didn't realize you were that close to the end.
Yeah, that's okay.
All right. Anybody have any particular questions? That was a tour de force. I don't know that I was aware that PACAP has such powerful effects, or the absence of PACAP has such powerful effects on energy expenditure and body weight. I mean, that's truly dramatic.
So, yeah, it was surprising to me. So, I mean, that wasn't my original hypothesis, but It was a happy accident that that there is the and this is something that is with each cohort, it's very reproducible.
Right. As a neuronal Neanderthal, is it as simple as if you give more PACAP centrally that you can induce energy expenditure and weight loss? Is it that simple?
So you can, that has been studied. The PACAP has been investigated for quite a while of the pharmacologic introduction, but what halted a lot of those studies were the genetic knockouts led very mice that were very sick and difficult to take care of, mainly due to the temperature regulation in the mice. They weren't able to handle carbohydrates, and that kind of led that research down a dead end. But can you administer just the PACAP itself into the CNS in order? I mean, people have done that. It suppresses food intake and increases energy expenditure and increases blood glucose. The problem with it, unfortunately, with a lot of these things in the brain is that, as I showed within the first slide, it's in the preoptic area, it's in the medial amygdala, it has roles in reproduction. So in and of itself, I'm not convinced is the key regulator, but potentially there's some way to tap into its ability to function that can promote energy expenditure.
And one last quick question from Maxence. Naturi, please.
Yeah, hey, this was a great talk. I really enjoyed it. One thing I was wondering is this protein ADCY-AP. So which adenylyl cyclases does this associate with and regulate?
That's a good question. I'm not familiar, at least on the signaling mechanisms that mainly I've been focused on the VPAC1 and the PAC1 receptor, which are G protein coupled receptors that that PACAP primarily acts on, but the actual signaling mechanisms that it engages, that I'm not completely sure, other than it tends to facilitate glutamatergic transmission, which at least that's where my mind is right now. And that what it's doing is it's just facilitating glutamatergic transmission when needed in response to leptin, T3, and glucose. But I mean, a lot of this stuff is, I'm in the early stages of trying to define what are the signaling mechanisms of all this. But overall, that I'm not sure.
Okay.
All right, thanks very much, Ebony and Jonathan, for two wonderful talks. Let me restate again that we will meet next time, February 2nd, Wednesday at 2 p.m. And we're going to do a talk from Michael Stitzel, a scientific talk, and then have a discussion of the future of the Pathways program, both in terms of plans for the ADA Pathways Symposium, but also plans for activities of our group going beyond the last currently scheduled meeting on March 2nd. So please join us both for the discussion and Mike Stitzel's scientific presentation. Thank you all and have a great afternoon. Bye-bye.